On this edition of the GARNet and INDEPTH Research Roundup, I'm really delighted to be joined by Professor Hank Bass from Florida State University. And we're going to be talking about a paper that's just been published in the Journal of Cell Science, the title of which is Identification and Characterization of Genes Encoding the Nuclear Envelope Link Complex in the Monocot species ZMAs. So Hank is a member of the INDEPTH Consortium, which is a EU cost program. The full name of the INDEPTH stands for Impact of Nuclear Domains on Gene Expression and Plant Traits. And this paper very much sits within the wheelhouse of that consortium. So thanks very much, Hank, for joining us today. And it would be great if you could give us an overview of the paper, please.
Well, thank you very much for the opportunity to talk to you about this. Yeah, so this is a big study that we've been chipping away at for a long time, and it all finally came together in the last year or so, led by my doctoral student, Hardeep Gumber. And so to give you a little bit of background for what brought us into this, we've been fascinated by the telomere nuclear envelope connection that occurs in meiosis. And this is widely conserved across eukaryotes. And it's probably the longest known and clearest example of the role for the link in a fundamental genetic process involving forces that move chromosomes around. And we've been studying this in maize by looking at mutants and 3D fish with telomeres, but we've never really studied the nuclear envelope during meiosis, and we wanted to do that. So that was a sort of underlying motivation to start this off. Okay. And we know telomeres move around like crazy, but they don't have motors per se. So the link complex was implicated. And at the time, we only really had two components in that machinery. We had the sun domain proteins known and an antibody to one of them, and the telomeres. But the rest of the complex was unknown to us. And Hardeep wanted to do a biochemical purification, but it's hard to get enough meiotic cells. And so we decided then let's get the link complex from somatic tissue, where we have an abundance of tissue. So our favorite tissue in maize is the ear shoot, the developing ear shoot. It's full of actively dividing cells, lots of nuclei, easy to extract. And so we started there by looking at proteins that co-purified with SUN2. So we have an antibody to SUN2. We did co-immunoprecipitation and started pulling out items. And of course, with these pull downs, you get a big mixed list of things. And Hardeep refined this and revised this quite a bit until almost nearly the 50th attempt. We started getting pretty good at it. And then the other sort of big breakthrough for this paper was working with the Oxford Brookes University group, Katja Graumann, Joe McKenna, and Francis Tolmie. And so what we did was, once we found a bunch of candidates, either informatically or by pull down in the co-IP, we then started making constructs with fluorescent proteins to put them back into plant cells and ask, do they go to the envelope? And if they do they show signs of interaction with sun, at least for the cache proteins? So that's kind of what the paper did. We ended up identifying 22 genes that we think are part of the link complex. And so there's the core complex, which is the sun and the cache domain proteins. And then there's everything else that they are connected to, either in the cytoplasm or the nucleoplasm. Once we had this, then we're sort of set to go back in future studies to look at the meiosis. So that's kind of the background and how this whole thing happened.
Okay, so you kind of set up this study's almost setting up for your future, your real interest, maybe.
It is, yes. We kind of hit a wall with the maize genetics. We had some mutants, and it's not easy to make mutants and make a lot of transgenic plants in maize. You can make them if you need them, but you can't make dozens of them to quickly test stuff. And so we've been looking for mutations in this pathway. There's a few here and there, and we decided we just need to get the complex identified and start from there. So that's the goal now.
So you mentioned pretty impressively that you tried like 50 different attempts for this IP, but in the paper you describe a slight kind of tweak of the protocol that you used in order to get the better results. Can you quickly describe what you did differently in that?
Yeah, I think probably the biggest breakthrough that Hardeep got worked out was using a study with a crosslinker called DSP, which is a short chain crosslinker. Because the challenge is you have to crosslink the complex, but you also have to leave the sun epitope sufficiently exposed for the antibody to get it. So there's a trade-off between locking up everything you want to co-purify and having access to an inner nuclear membrane epitope that's between the two membranes of the envelope. And so, there's a really narrow sweet spot in there with regard to fixation and not fixed. And so this chemical is called DSP, and it's from a study from Jaffarali et al., 2014, and they refer to this method as MCLIP, M-C-L-I-P, and it's membrane protein cross-link immunoprecipitation. And what was exciting about that study was that they had shown they were able to recover sun-interacting proteins in U2OS cells. So that gave us some confidence that we could apply this to plant cells, and that really helped sort of clean up. And we already had our list of candidates, so we knew what to look for. We're looking for cache proteins, we're looking for the crown proteins, and so we were looking for those in particular, and we saw a few other things, but I think that was probably the biggest breakthrough.
So you identified this suite of proteins, and as you mentioned, then you, in collaboration with the guys at Oxford Brookes, you actually did some microscopy and some FRAP as well to show that these proteins were localized to the nuclear envelope. So can you just describe a little bit about those experiments that you did?
Yeah. So the most challenging members of the link complex are probably the outer nuclear membrane proteins, the cache proteins. They don't have a lot of diagnostic domains. They have a transmembrane domain, a short 2 or 3 residue termini that interacts with sun, but there's not a lot else to identify them. So we wanted to test those, and so we made GFP fusion proteins and then introduced those into tobacco, Nicotiana benthamianum, as a sort of test case. So this is a monocoque grass-specific gene going into a eudicot species, and in the leaf epidermal cells, you can then look within 48 hours of a transit transfection. You can look and see where the protein is, and so one of the primary expectations was if these are bona fide link complex proteins, they should go to the nuclear envelope. And so, with no other evidence, we started just looking at those, and so that. Once we could see that they did go to the nuclear envelope, they also seemed to go to the ER quite a bit and other membrane systems. And so we started knocking out domains to see if they had the sort of expected C-terminal requirement for nuclear envelope localization and interaction. And then in the FRAP experiments, those gave us really good evidence for interaction with sun via sort of indirect interpretation of change in mobility. Figure 6 shows the fluorescence recovery after photobleaching, FRAP experiments. And the way this is done is that the cache protein with the GFP is expressed you can see it in the nuclear membrane area, and then it's bleached, and then the fluorescence returning to that area is measured over time. And so when it returns, only a certain proportion of it returns, and that tells you what proportion of the protein is mobile versus immobile. So you get a baseline mobility. And then if we co-express with maize sun, so now we're expressing maize cache and maize sun in the tobacco system. If you overexpress with maize sun, you get an increase in the amount of the immobile fraction. So more of it is immobilized. So we took that as indirect evidence, as has been previously published by Katja and their group, that sun and cache interact.
I'd urge people to go and look into the supplemental, because there's some really nice representative images within the supplemental data showing the nucleus and showing the nuclear envelope and these FRAP experiments. Okay, so just as a final question for you, Hank. So where are these experiments leading on to? So you've got really this nice baseline of experimental data now from the maize system. So what are you planning over the future in the future?
So one of the big questions going forward is which of these proteins give specificity to different tissues and processes. So we've kind of done the whole set collection. These are the 22 most likely fundamental proteins in the link complex, but they're not all expressed in all cell types. So we're starting to look at tissue-specific expression. For example, now we're working on the SIGN2. The SIGN genes have armadillo domains that appear to interact with actin cytoskeleton. So those are really interesting. Also, if you look at the figure with the conservation.
Mm-hmm.
What's interesting is that, and that's figure 3, there are 3 classes of the links that are conserved all the way out to bryophytes. They are the C-terminal sun domain proteins, the NMCP crown, which we call NCH, and the MLKS2, the sign-like cache proteins.
Mm-hmm.
Those 3 groups might represent the core nucleoplasm to cytoplasm set. So we'll probably be focusing on some of those. Any of the ones that are expressed in meiosis, we're going to look at now and see, is the telomere bouquet disrupted? Is chromosome pairing and recombination disrupted? Do they need these proteins to get through the meiotic chromosome segregation phenomena that demand so much on the nucleoplasm-cytoplasm interaction?
Cool. All right. So thanks very much, Hank,
Thank you.
for discussing the paper today. And as I always say, we look forward to learning about those outputs over the coming years.
Thank you very much.